This will get technical, but good technical. DNA breaks all the time. You go get a dental x-ray. The technician points this thing at your face, and the x-rays actually hit your DNA, and they physically create a break, so that the familiar double helix of DNA physically goes. The good news is, the cell has its own machine to fixing breaks. Inside our cells, there are two identical DNA molecules lying side by side, literally side by side. If one is broken, it can say to its sister, and that in fact is the technical term, the sister, "Hey sis, I'm sorry, I've had a break. I'm wondering if I can copy the missing genetic information." And the sister goes, "Yeah." Done. Why is that useful? So it's useful because if you can cut a gene inside a cell, so if you can create a break at a place of interest, then you can change that gene. You fool the cell. Give it a separate piece of DNA that you have made. A piece of DNA which is identical to the chromosome that you're cutting, except for the change that you wish to make. And Mother Nature will not know she's being fooled. She will repair the break using this piece of DNA you provided as a template. And so whatever change you brought in will then go into the chromosome.
So if you wanted to use that capability to engineer the genome, the challenge was to introduce breaks in the DNA at places where you wanted to alter the code.
How are we going to do that? We need something that cuts only one gene out of the, 25,000 that we have.
There were just such serious blocks in the way. So it looked like it was going to be a long road. And that's what changed. And that came sort of overnight.
When Cas9 polices the intracellular neighborhood for invasions, it literally carries a copy of that most wanted poster with it, asking everyone that comes in, "Excuse me, do you contain an exact match to this little most wanted poster that I'm carrying? Yes? Then I'll cut you." In which they describe that Cas9 can be directed. Cas9 cuts DNA based on an instruction that it carries, and that instruction is a molecule of RNA that matches perfectly the DNA of the invader.
RNA, I think about it as DNA's chemical cousin. Like DNA, it has 4 letters, and they can form pairs with matching letters in DNA. The letters in the RNA allow Cas9 to find a unique DNA sequence. Bacteria are programming this thing all the time with different viral sequences and then using it to find and cut and destroy those viruses. But because it's using these little RNA molecules, those can easily be exchanged. RNA molecules are trivial to make in a molecular biology lab or order from a company. It actually allows us to change human evolution if we want to, right? It's that profound.